Hello, welcome to your online lecture for the course Anatomy and Physiology 2. In today's lecture, we are covering the immune system. Our immune system is designed in such a way that it can protect our body under many circumstances against physical attacks on it, whether it be something that's happening externally onto the body or internally onto the body. But the key with our immune system is cell identification. In other words, our body being able to identify what is normal and what is abnormal. Maybe it'll attack what's abnormal, but we certainly don't want it attacking what is normal to our body. Let's first define the term antigen. Cells in our body, viruses that come in our body, and other particles, they have these unique molecules and groups of molecules that are found on the surface of themselves in order to identify them. These molecular markers that are visible to our immune system are called antigens. So you can think of an antigen as being a cell identity tag that says who and what that cell is. Our own cells have unique cell markers as well, or antigens, that are embedded within the plasma membrane of each of the cells in our body that identify it as being self, that is, belonging to us as the individual. Foreign cells and foreign particles have these non-self molecules or non-self antigens, and they serve as recognition markers for our immune system so that our immune system can look at these particles and say, well, that's not normal, so this is something that we need to attack. There are two different types of immunity that we're going to discuss: innate immunity and adaptive immunity. Innate immunity, oftentimes called non-specific immunity, these are mechanisms that are built into our body and they're ready for action. They're really providing us with our initial defense against infection and other assaults on the body. Whereas adaptive immunity or specific immunity, these mechanisms develop later. They adapt. Your body adapts. That's why it's called adaptive. And they develop because lymphocytes, a type of white blood cell, are activated to work against specific foreign or abnormal cell products. We're going to be speaking about these different types of immunity in a lot more detail throughout the rest of today's lecture. So after the lecture, it would be a good idea to come back to this slide because you'll then understand the components better. Let's talk for a moment about lines of defense. Think of an army as part of the war, and you have your first line of defense, which is going to be at the front, and then you have a second and third line of defense behind that. Okay, immune function. That is defense of the internal environment against foreign cells and proteins, and viruses include 3 layers of protection: the first, second, and third line of defense. The first line of defense is a set of barriers between the internal and external environment. So, for example, your skin. The second line of defense involves innate inflammatory responses, including the process of phagocytosis, which you'll learn a little bit more about later. And the third includes the adaptive immune response and the innate defenses offered by, in particular, natural killer cells. Of course, tumor cells that arise within the body are not affected by the first 2 lines of defense, and so if our body develops cells that are abnormal within us, in other words, they didn't come from the outside, they developed within us, then they will be attacked, hopefully successfully, by our third line of defense. And so here our first line of defense, being our skin, has been broken. And so now we need the second line of defense to pick up. So whatever is entering and has bypassed our first line of defense is now in our body, and this is where our second line of defense, so we have these larger cells called macrophages that can go through a process, partake in a process called phagocytosis, where they can consume and digest anything that's foreign coming into our body. But then in some circumstances, a third line of defense picks up and our body is going to produce antibodies against whatever came into the body, so that should it come into the body again, we'll be able to fight it off faster. Species resistance refers to a phenomenon in which the genetic characteristics common to a particular kind of organism or species provide defense against certain pathogens. Pathogens are disease-causing agents. We, as humans, are resistant to certain funguses that infect trees, for example. The internal environment of the human body is protected by a continuous mechanical barrier that's formed by cutaneous membranes, so really the skin, as well as mucous membranes. Often called the first line of defense, these membranes are going to provide several layers of densely packed cells and other materials, forming sort of a wall that protects the internal environment from invasion by foreign cells. Besides forming a physical protective wall, the skin and the mucous membranes operate various additional immune mechanisms as well, these chemical barriers. Substances such as sebum, which contains pathogen-inhibiting agents, mucus, in which pathogens can become stuck in and end up being swept away, enzymes, which may hydrolyze pathogens, as well as hydrochloric acid that's found in gastric or stomach mucosa, which can destroy pathogens. All of these may also be present to act as part of our innate defense. If bacteria or other invaders break through the chemical and mechanical barriers that are formed by the membranes and their secretions, the body has our second line of defense that now kicks in, and this is referred to as the inflammatory response. Tissue damage elicits this host of responses that work to counteract the injury and really promote a return to the normal state. So, for example, bacteria, they can cause tissue damage that will then in turn trigger the release of various inflammatory mediators from cells such as mast cells, which are found in connective tissue throughout the body. Inflammatory mediators that are released from these mast cells include histamine. Other cells release cytokinins and prostaglandins. But in any case, these mediators are working to dilate blood vessels and increase vascular permeability. And this has a purpose, which is to help phagocytic white blood cells reach the affected tissue and get to work. Some of the characteristic signs of inflammation include heat, redness, swelling, and pain, as well as loss of overall function of that area. Many of the mediators of inflammation that are listed above here are chemotactic factors. That is, they're chemicals that attract white blood cells to the area in a process called chemotaxis. Let's have a look for a moment at both chemotaxis and something called diapedesis. In this example here, we have an injury. So this is the site of injury where inflammation is occurring. A neutrophil, represented here, is attracted to the area by chemotactic agents released by mast cells that are found in damaged or infected tissue. So here's a mast cell. It's going to give off these chemotactic factors, almost like a flag for help, a signal for help that attracts neutrophils to the area. Neutrophils can exit the capillary, which will become more permeable, exit the capillary by a process called diapedesis. And then they can enter the tissue. And through the process of chemotaxis, the neutrophil is able to migrate to the highest concentration of this chemotactic factor. So it's basically following the chemicals, which is the site of injury. And this is where it can then begin its immune functions. As you should recall, neutrophils have phagocytic functions. You should also recall that diapedesis is the process by which certain cells then can pass through this uninjured wall, because this vessel isn't necessarily injured, but it can still squeak through and enter tissue where it can start cleaning up. A major component of the body's second line of defense is the mechanism of phagocytosis, which is the ingestion and destruction of microorganisms or other small particles. There are many types of phagocytes that exist within the body. Again, these are cells that are capable of phagocytosis. And this image summarizes the function of a phagocyte. Here's a phagocyte here, and this type is a macrophage. When a phagocyte approaches a microorganism, so here's the microorganism, it will recognize it as not being normal. It will then extend these foot-like projections called pseudopods toward it. Soon, the pseudopods will completely encase or encircle the organism and form a complete sac. This is called a phagosome. The phagosome then moves into the interior of the cell, or the interior of the macrophagy, where it will come into contact with lysosomes found already inside the cell. And so you can see those lysosomes are fusing now with the phagosome, and it ends up getting the name phagolysosome. The contents of the lysosome, so within a lysosome, we have these very, very potent digestive enzymes that are going to break down the contents of that phagosome, and it will destroy the microorganism within it. And then eventually, through the final process here, is it will release its digested contents and our body will be able to clear it out. And then it's able to repeat that cycle. Many cells are capable of phagocytosis, but the most numerous is the neutrophil. And this is the cell that's usually the first to arrive at the site of injury. Think back to our image a couple of slides ago, and it will migrate out of the bloodstream through the process diapedesis. In addition, neutrophils are involved in the formation of pus. Because phagocytosis defends us against various kinds of agents, it is classified as part of our innate defense, but it does also cross over into adaptive immunity as well. Now, thinking back to the last slide, after the phagocyte digested the offending particle, the phagocyte, or the macrophage, will process proteins from this offending particle, then take those proteins, we'll call them peptides, and display bits of it on the surface of itself. So think of it like the macrophagy attacks the foreign particle, starts to digest it, and then takes some of it and wears it, puts it on its surface. These peptides can then be recognized by cells of our adaptive immune system as antigens. Cells that perform this function are called antigen-presenting cells. Another common type of phagocyte that we've already talked about is the macrophage, which means large eater. Macrophages are large monocytes that have grown to several times their original size after migrating out of the bloodstream. They are important antigen-presenting cells. Yet another type of phagocyte is called the dendritic cell, which is found in many tissues of the body that have contact with the external environment, so such as the skin and mucous membranes. And it's called a dendritic cell because of its many branches. Dendrite means branches. If you think back to nervous system anatomy and those dendrites off neurons, those branches. We've spoken a bit about local inflammation, but systemic inflammation may occur when inflammatory or inflammation mediators trigger responses that occur on a body-wide basis. A body-wide inflammatory response may be manifested by a fever, which is a state of abnormal high body temperature. So for example, bacterial or viral infections that spread widely throughout the body may produce what's called systemic inflammatory response syndrome, or SIRS. On to the third point. It is pyrogen molecules that trigger this fever response when it is released from damaged tissue or when it's introduced into the body, such as a bacteria. Endogenous refers to originating within the body. Exogenous refers to originating outside of the body. So pyrogens then induce fever. Pyrogen molecules do this in a particular way. They trigger the fever response by promoting prostaglandin production. Remember that from one of our first lectures. Prostaglandins reset the body's thermostat within the hypothalamus, and they set it to a higher temperature in order to burn off the pathogen, so to speak. Aspirin and other cyclooxygenase inhibitors, or COX inhibitors, can reduce the production of prostaglandins. And when you reduce the production of prostaglandins, and they're no longer turning the thermostat up too high, then the body temperature can be brought back to normal. And this is why many medications taken when you have a fever can make you feel a lot better, because fever comes with symptoms such as aches and pains, fatigue, etc. It's thought that the elevated body temperature is designed to facilitate an immune response and possibly increase immune function, but the role over fever and whether or not it's a good thing or a bad thing and to what extent is still being debated among immunologists. Besides phagocytes, the body has another important set of cells that provide innate defense in the body, and these are called natural killer cells, or NK cells. NK cells are a group of lymphocytes that kill many types of tumor cells and cells that are infected by different types of viruses. Natural killer cells recognize abnormal cells by using two recognition receptors, a killer activating receptor and a killer inhibiting receptor. If we look at this diagram here, this is representing a natural killer cell, and this is a normal cell in our body. You can see that the natural killer cell, as just mentioned, possesses a killer activating receptor. So this receptor would be what would activate the killing of the cell that it binds to, but it also has a killer inhibiting receptor. So let's talk about each. The killer activating receptor binds to any of several common surface molecules that are found on cells. So for example, this typical surface molecule here. Thus, the natural killer cell then can bind to any cell of the body as well as foreign cells. However, if the killer inhibiting receptor happens to bind to what's called MHC protein, which stands for major histocompatibility complex, then the killing action is stopped. So you can see that the killing activator receptor bound to a normal cell in the body, but because it also bound to a very specific receptor bound by or to the killer inhibiting receptor, then the killing action is stopped. So this is where the natural killer cell is sort of indirectly realizing that this is not a foreign cell. MHCs, or major histocompatibility complexes, are surface proteins on all normal cells and are unique to each individual person. So, normal to you. Only abnormal and foreign cells fail to bind to that killer inhibiting receptor and are therefore killed by a natural killer cell. So you can see in this example here's an abnormal cell. The killer activating receptor binds to its surface molecule, and the killer inhibiting receptor does not bind to anything, so the natural killer cell gets the go-ahead to kill that cell. The natural killer cells use several different methods for killing cells. Most of them involve or progress to what's called lysis, or the breaking apart of that abnormal cell so that it's no longer functional. But in any case, direct contact by the natural killer cell to the abnormal cell is necessary for it to be broken apart or lysed. Several types of cells, if invaded by viruses, so we're talking about cells in our body, if they're invaded by a virus, they can respond rapidly by synthesizing a protein called interferon and releasing some of this interferon into your circulation. The interferon that is released to nearby cells in your body is going to signal those cells to enter into a protective antiviral state. In other words, warning signals are sent out to the other cells to help prepare for this viral attack. So one cell gets attacked, it's kind enough to send out a warning to all the other cells in the body. As the name suggests then, interferon proteins interfere with the ability of viruses to cause widespread disease within the body. Complement is the name given to each of a group of inactive enzymes that end up being activated in a cascade of chemical reactions, which will end up resulting in a variety of different immune responses. Ultimately, the complement cascade is going to cause lysis or rupturing of foreign cells. Complement proteins essentially complement or aid in the immune response. There's a lot more detail on the complement system than what we go into in normal anatomy and physiology, but in other courses, altered physiology, pathophysiology, that's when you'll learn a lot more about this complement cascade. And so we can collectively refer to both interferons and complement proteins as being protective proteins. We have just finished discussing innate immunity, and now we'll move on to talking about adaptive immunity. Adaptive immunity is considered our third line of defense. This type of immunity is specific rather than non-specific like innate immunity. So there are specific cells that have adapted to kill specific microbes. Adaptive immunity is orchestrated by two different classes of white blood cells called lymphocytes. The two major classes are B lymphocytes or B cells and T lymphocytes or T cells. And as we'll discuss shortly, B cell mechanisms are oftentimes classified as antibody mediated immunity or humoral. Humoral refers to fluid. T cell mechanisms are often classified as cell mediated immunity or cellular immunity. Now, relating to this image here, recall from one of our previous lectures that lymphocytes originally form in bone marrow, as do all blood cells, from these primitive cells called hematopoietic stem cells. Now, T cells, they actually travel to the thymus to mature. So think of T for thymus and T for T cells. B cells, however, they're going to mature primarily in the bone. So think of B for bone cells. And so this will happen before it ends up entering into circulation, where it will then travel to both the T and the B cells, where it will then travel to lymph nodes as well as the spleen, where they'll be involved in our immunity. Adaptive immunity requires activation of B and T lymphocyte populations, which then begin their immune attack of specific antigen after they're activated. But such activation requires two activating signals: a specific antigen and a chemical signal that's released from damaged or infected cells or from the attacking microbes. You can see here then that each lymphocyte is going to have receptors for antigens and for the signaling chemical in order for activation of immune cells to occur. As I've already told you, B cell immunity is also referred to as humoral immunity, and that B cells develop in and start to mature in red bone marrow. At this point, though, they're referred to as naive B cells. And you could say that this would be after completing their first developmental stage. These naive B cells, they synthesize antibody molecules, but they secrete very few, if any of them. But what they do instead is they insert many of these antibodies that they produced onto their cell surface, so onto their plasma membrane. Once released from the bone marrow, these naive B cells are going to travel to places such as the lymph nodes, they're going to travel to the spleen, and even to the liver. In order for these naive B cells to be activated, though, they must first have an encounter with a very specific antigen that fits perfectly with the antibodies that it's placed on its cell surface. When the antigen binds to these antibodies on the B cell surface, so something foreign has a perfect bind, then activation of the B cell occurs. And this will then trigger a rapid series of mitotic division, essentially creating a clone of itself. The clone will then differentiate into many different types of cells, such as plasma cells and memory cells. Plasma cells synthesize and secrete huge amounts of antibodies, and we're going to talk about specifically what antibodies are on the next slide. Also called effector cells, memory B cells, they do not secrete antibodies, but if they're later exposed to the antigen that initially triggered their formation, then they will rapidly divide rapidly divide in order to produce more plasma and more memory cells. So what are antibodies? Antibodies are a protein of the family called immunoglobulin, and you'll see that written as Ig. Like all proteins, antibodies are very large molecules, and they're composed of a long chain of amino acids called a polypeptide. So think back to learning about transcription and translation and the formation of a polypeptide or a protein at the level of the ribosome within the cell. Each immunoglobulin molecule consists of 4 polypeptide chains, 2 that are heavy and 2 that are light. The chains are folded to form these globular regions that are joined together in such a way that the immunoglobulin molecule as a whole takes on this Y shape. You can see in the diagram the 2 twisted red strands, which are considered the lighter chains, the light chains, and 2 twisted blue strands that are the heavy strands, or heavy chains. The heavy strands are longer and heavier than the light strands are. The immunoglobulins contain what are called variable regions. You can see that's written here, variable regions. And these are regions where the sequence of amino acid is going to vary in different antibody molecules. This is where the differences are going to be amongst different antibody molecules. This is represented by just the colored bars that you can see in figure B here. These regions have the ability to take on different shapes so that they can bind to different types of antigens. So different naive B cells, so different because they contain different surface antibodies, can then bind to different microbial antigens. The clonal selection theory deals with antigen destruction. And this theory holds 2 basic points. First, it holds that the body contains an enormous number of diverse clones of cells, each that are committed to synthesize a very specific antibody. Second, when an antigen enters the human body, it selects the clone whose cells are committed to synthesizing its specific antibody and stimulates these cells to proliferate and to therefore produce even more antibodies. And so I spoke about this when we introduced B cells and spoke about the plasma cells and memory cells a few slides back. This graph shows antibody response time. The initial encounter with a specific antigen, so the primary stimulus, will produce the primary response. And as you can see in this diagram, there's an increased production of immunoglobulin M and immunoglobulin G, and this will happen over the course of many days. You can see that the antibody levels drop off at some point, and that's because the antigen has been dealt with. Remember that memory B cells have been produced, and they're going to be waiting around now for some sort of subsequent exposure. Now let's look at a later encounter with the same antigen. This will trigger those waiting memory cells to act faster than before, and they will produce even more memory cells and plasma effector cells that release more antibody than before. And so this later encounter, or we can say the secondary stimulus, is going to produce a secondary response, but you can see that this is a much faster response and a much grander response. In particular, with immunoglobulin G, very large increases compared to what we saw with the primary stimulus and the primary response. So these IgM and IgG, these are very specific antibodies. So we're going to be spending some time now talking about the different forms of immunity. Natural versus artificial, and active versus passive. Natural immunity can exist in active or passive form. Active form would mean that the person is exposed to something and then they actively produce antibodies. So for example, someone in my household has a virus, I become infected with that virus, and then my body produces antibodies against that virus, actively producing antibodies. Natural, because I was just naturally exposed to it. Natural passive immunity, however, is where antibodies are given to you passively. And so the example is when a mother is breastfeeding. So she's breastfeeding. She is going to be able to give the baby antibodies. Instead of the baby actively producing antibodies on their own, they are passively acquiring them from the mother's milk. It's natural because it's a natural process of breastfeeding a child. Artificial immunity, however, is a little bit different. We can gain artificial immunity actively or passively. Actively is through immunization, so giving a vaccine. So the body is artificially, as opposed to naturally, coming into contact with a pathogen, artificially because it's being given to you in this pre-made injection. And so at that point, artificial injection of pathogens, exposure to antigen, then the body will still actively produce antibodies, but was exposed to it artificially through immunization. Artificial immunity can also exist in passive form. So remember, in the case of passive, you're not actually producing the antibodies yourself. So for example, let's say I throw something into the garbage in a public garbage, and I realize that I think I dropped my wallet in there as well, and I reach in and I prick my finger on a needle that was in the garbage. It's possible that that needle could be infected with some virus. And so instead of waiting for my body to produce antibodies, because that takes time, just in case it's a very serious virus such as HIV, doctors can actually inject antibodies into me. And so that's passive because my body's not actually producing the antibodies, but they're being directly injected into my body. And it's artificial because it's a pre-made substance being injected into you, compared to the previous slide when we talked about natural, because these antibodies are coming directly from the mother's milk. The function of antibody molecules is to produce antibody-mediated immunity, which we know is a type of immunity also called humoral immunity. I mentioned earlier that humoral refers to fluids, and this type of immunity occurs within the plasma, which is one of the body's humors or fluids of the body. Essentially, antibodies act on antigens by inactivating and bending them together to facilitate phagocytosis, and by initiating inflammation and activating the complement cascade. Remember, just as a reminder, complement is a group of enzymes that produce a cascade of reactions that result in a variety of immune responses. Now, with all that I've just said, let's look at that a little bit closer by using this diagram. As you can see in the diagram, we have an antibody, and it's going to either recognize this antigen as being self or non-self. And if it recognizes it as not self, then or foreign, then it will produce one or more effects. It may inactivate the enzyme, transforming it to something that's not harmful anymore to the body. It may also cause agglutination. Agglutination is the clumping, and in this case, the clumping of antigens together. And this will allow the antigens to be more easily accessible by macrophages and other phagocytic cells in order to perform phagocytosis. The binding of the antigen to the antibody still produces another effect in that it activates the complement cascade that we spoke about earlier that will contribute to the destruction of these foreign cells through many different processes, which leads to this complement cascade, inflammation, chemotaxis, and lysis. We learned earlier that mast cells are involved in the inflammatory response. Antibodies become attached to them, so here's the mast cell. An antibody can become attached to that mast cell and followed then by the antigens, which will initiate a release of inflammatory chemicals. We have spoken a fair amount specifically about B cells, but now let's elaborate on T cells. T cells initially develop in red bone marrow, then they mature at the level of the thymus, and at this point they'll migrate to lymph nodes and the spleen. When an antigen encounters a naive T cell, the antigen will bind to the T cell receptors. T cells have antigen receptors on their surface, but they're different yet similar to the ones that B cells have on their surface. This will end up causing activation or sensitization of the T cell, leading to rapid mitotic division into very specific sensitized T cells that we call effector T cells and memory T cells. Effector T cells are going to secrete a variety of different cytokines. Cytokines are a very broad category of proteins that are involved in cell signaling. And then finally, memory T cells, these will remain in the red bone marrow until they produce additional effector T cells when needed. So here are the memory T cells producing additional effector T cells when needed. T cells display antigen receptors on their surface membranes that are different than the antibodies, but similar. So remember that antibodies, when we talked about the B cells, antibodies are present on the surface of B cells. T cells, however, can only react to foreign antigens that have already been phagocytosed and then presented on the surface of antigen-presenting cells. This junction between the T cell and the antigen-presenting cell form this temporary junction called an immunological synapse. You can rewind back to my discussion on B cells and then maybe re-listen to the T cell component so that you can understand the main differences between them. Of the effector T cells, so we really generally stated effector T cells. Of the effector T cells are cytotoxic T cells, which cause contact killing of a target cell. You can see in this diagram on the right that cytotoxic T cells, also referred to as killer cells, are attacking cancer cells. So here we have the cancer cell, and here we have the cytotoxic T cell. T cells are a significant part of our defense against cancer and other abnormal or foreign cells. After forming an immunological synapse, which you can see happening here with the tumor cell, the cytotoxic T cell releases what's called perforin, which essentially triggers apoptosis or cell death of the cancer cells. So perforin, it almost perforates the membrane in order to allow for granzymes to then enter the cancer cell and attack or trigger apoptosis. This figure summarizes the stages of the adaptive immune response. First, B cells and T cells recognize a specific antigen. Next, the B and the T cells become activated, expanding their population by cloning, while differentiating into effector cells and memory cells. Then the effector cells get to work by attacking the source of the antigen. Both antibody-mediated immunity and cell-mediated immunity will take place. As antigen levels decline, effector cells will die off through this pre-programmed cell death, and then the memory cells remain. And the memory cells will remain to be ready to very quickly engage and be prepared for the antigen if it does enter the body again. That's the end of today's lecture. Thanks for listening.